Welcome to Unit 3 of AP Biology, Cellular Energetics. In this unit, we're going to be studying photosynthesis and cellular respiration, as well as some background leading up to those. So this video covers 3.1 and 3.2 of AP Biology, which is Introduction to Energy, Enzyme Structure, and Catalysis. So let's start with a basic question: what is energy? Energy is simply the capacity to cause change. So here in this diagram, we have three different examples of change that might happen as a result of having energy. So you can have gravitation, you can have diffusion, or a chemical reaction. In all of these cases, if this is a spontaneous change, we're moving from a state of more free energy, which is less stable and has more capacity to do work, to a state that is more stable and therefore has less free energy and therefore less work capacity. So we're going from a position of higher energy to lower energy in all of these pictures. So as I mentioned in that last picture, a spontaneous reaction is one that does not require a net input of energy. So take a look at these energy diagrams and figure out which graph shows that. So the graph on the left is showing a spontaneous reaction, one that does not require a net input of energy, and in fact, in exergonic reactions, energy is released. And you can see that because the energy of the products in an exergonic reaction is lower than the energy of the reactants. So we always want to be looking for the reactants and the products in terms of whether a reaction is spontaneous or not, because that's sort of the difference that really matters, is looking at the before and the after. You're always going to have a higher energy level is the intermediate, because it's always going to take more energy to get to that transition state. And that amount of energy that it takes to get from the reactants to that transition state is called the activation energy. So a couple more terms to be aware of. Delta G is the change in energy. So G stands for Gibbs free energy. You need to understand this concept in general, but you don't need to do any mathematical computations for AP Biology related to calculating free energy from delta H or delta S. You may get to that in chemistry. So delta G gives you an indication of whether a reaction is spontaneous or not. If delta G is less than 0, it means it's spontaneous, because in this case, energy must have been released. If delta G is greater than 0, that means you needed to put energy into the reaction, because the products have more energy than the reactants, and that is not a spontaneous reaction. So let's look at why this is. So in this diagram, does this show an exergonic or an endergonic reaction? To figure that out, what I would want you to look at is that the most basic, we're looking at going from reactants to a transition state to products. Going from reactants to transition state, we always have to take in energy, because that requires energy to break those bonds. Going from transition state to products gives out energy, because we're making new bonds, so it's becoming more stable, has less energy. So to determine whether it's exergonic or endergonic, you can really ignore that transition state and just compare the reactants to the products. So are we needing to put net energy in, or is energy released? And in this case, the energy level of the products is lower than that of the reactants, and therefore there's a net release of energy. So in this case, this is showing an exergonic reaction. All right, so when we're talking about energy, it's important to understand enzymes and what enzymes do. First of all, why is it important to talk about enzymes when we're talking about energy? And second, what's happening in this diagram? What is the enzyme actually doing in each of these steps? So the reason we want to talk about energy and enzymes together is that enzymes are going to lower the activation energy of a reaction. We'll come back to that in a little bit, but let's take a look at what the enzyme is actually doing. So the enzyme is providing a sort of environment where the reactants can come together to create the products. So when we're talking about enzymes, the words substrates and reactants can be used interchangeably. So the first step is those substrates enter the active site. The active site is the area of the enzyme where those substrates fit in, sort of where the action is. The second step here is that substrates are held in the active site by weak interactions. What it's showing third is that the active site can lower the energy of activation and speed up a reaction. And this is what I was saying, why enzymes and energy are so important to be talking about together. The fourth step here is showing that substrates are converted to products. So here out come the products. That's step 5, the products are released. And then the sixth step is just indicating that now we're ready for the first step again. So the active site is available for new substrate molecules. So you'll notice that the enzyme is not destroyed in this process. The enzyme can continue to facilitate this interaction. All right, now we've got another diagram of enzymes. So here we have enzyme and substrate, and then ES complex. What's going on in this diagram? So something I want to point out in this diagram, first of all, ES stands for enzyme substrate complex. That's when they've come together. But I want to make sure you understand that the enzyme is changing shape a bit. So the enzyme and its active site slightly change shape to allow for a complete binding of the substrate. This is called induced fit. So you can see the picture of the enzyme before and after binding with the substrate is a slightly different conformation, slightly different shape. Something else to keep in mind is that not every enzyme will work with every substrate. There is a real specificity, so only the correct substrate can induce the proper alignment of the active site that will enable the enzyme to do what it's supposed to do, to perform its catalytic function. All right, let's take a moment and sort of get a little off topic, and then we'll get back onto topic again. So I don't know about you, but I love silly cat videos, and there's a great silly cat video out there of this family who built an amazing Jenga tower, and you can see that Jenga tower over on the right there. And in this cat video, that cat pictured in the photo there investigates this Jenga tower so much that it actually gets inside, and you can imagine what follows when the cat gets inside the Jenga tower. So from this video, I want you to remember that Anna builds things. So she's this girl pictured here building an amazing Jenga tower, and the cat breaks them. So let's see if we can make any sense of why I just told you a story about a cat. So take a look at these two diagrams and tell me what's another name for a synthesis reaction and a breakdown reaction. So the name for a synthesis or building reaction, where you have two parts of the substrate coming together and making a larger molecule, this is called an anabolic reaction. A breakdown reaction, where you have a larger molecule breaking into smaller components, is called a catabolic reaction. So if you remember from our silly cat video, Anna builds things, cat breaks them. A few more definitions for you. Cofactors and coenzymes. So what are they in general? So there's something that defines both of them together. And then what are they independently? How would you define them differently? Cofactors and coenzymes are both helper molecules that activate enzymes. They are different in terms of their composition. So cofactors are small inorganic compounds and ions, whereas coenzymes are non-protein organic molecules. So this brings up the question, what's the difference between inorganic and organic molecules? And how can you identify organic molecules? So generally speaking, organic molecules have oxygen and hydrogen and are found in living systems. So a follow-up here is, what are the 5 types of organic molecules that we discussed in Unit 1? So the 5 types of organic molecules that we discussed in Unit 1, proteins, lipids, carbohydrates, nucleic acids, and hydrocarbons. And if you need a review of those, please go back to some of the Unit 1 videos. All right, here we go, continuing with some more definitions. Definition of the term catalyst. So what does this mean outside of biology? It means a person or a thing that precipitates an event. See if you can come up with an example of a catalyst. There are lots of right answers here. And then how would you define a catalyst within biology? So one example of a catalyst, and like I said, there are many, would be the idea that fortunately for battery manufacturers, the ice storm was a catalyst for increased battery sales. So there's something that precipitated an event. So the ice storm precipitated the battery sales. So it sort of created it, it inspired it. Within biology, the definition of a catalyst is a chemical that increases the rate of a chemical reaction without itself being changed by the reaction. So now let's put this together. We just talked about what a catalyst is. How does this apply to enzymes? What's the meaning of the root word cata, and why is an enzyme said to catalyze reactions? And then a little review. Take a look at this energy diagram and tell me if it's an endergonic or exergonic reaction, and how can you tell? And then take a look at the effect of the enzyme, and so draw the other type of reaction, the one that's not pictured here, both with and without an enzyme catalyst. So one of the definitions of the root word cata means down. So you can think about that as we were applying it to catabolic reactions, is that the catabolic reactions are breaking things down. When you're applying it to catalysts, you can think of enzymes as bringing down the energy of activation. So in a reaction, you can see in both of these energy diagrams, a reaction without an enzyme has a much higher energy of activation. That's that change between the reactants and that transition state. When there is an enzyme, it takes down that energy of activation. All right, some follow-up questions here. Are anabolic reactions generally exergonic or endergonic? And how about catabolic reactions? Are those generally exergonic or endergonic, and why? So remember, anabolic reactions generally build things up. It's generally going to take energy to build things up because you are putting them into a less stable state. So it's going to require a net input of energy. So anabolic reactions are generally endergonic, as opposed to catabolic reactions, which are breaking things down into simpler molecules. Those are generally going to be exergonic reactions because the energy of the products are lower than the energy of the reactants. They're more stable and therefore have less energy. So that's it for this video. In the next video, we will dive a little bit deeper into enzymes and look at what factors affect enzyme behavior.